Last but not least of the marathon today. So we have Jonathan, Cardenas Galego. So Jonathan had graduated in 2019, right? Jonathan graduated in 2019 from the university. So let me say the right name: Universidad San Francisco de Quito. There you go. In Ecuador. And Jonathan, like me, is an agronomic engineer. So Jonathan had joined the program about 2 years ago. And he built on what Mark just presented. So we collaborated with Mark Vanier. So when they used the RGB camera and added a lot of other nice gadgets into it. So today we're going to see the summary of everything that Jonathan had done. So Jonathan, the floor is yours.
Yep, thank you, Ronito. Everybody on Zoom, can you hear me? Can you see my slides? You can. I don't hear any complaint.
Looks good.
Looks good. I'll start. So hello, everyone. I know it's been quite an exhausting day for us, 3 presentations in a row, but thanks, Mark. You set the stage for imaging now. It's going to be the portion that I'm going to be covering and what I've been working during these 2 years. So Mark started working with germination. Now I will follow the next part of the crop cycle as we try to see if we can estimate accurately biomass and also provide tools for tipburn monitoring within a greenhouse. I would like to start talking about Okay, I would like to start talking about plant phenotyping. Plant phenotyping initially was introduced to bridge the gap between what the genomics development was, and also to try to find an interconnection between the genotype and environment interaction, as we were pretty limited with the phenotypic characterization doing manually, traditionally. Now, initially, it was envisioned for breeding and stress resilience problems like fundamental research. However, phenotyping has developed in a really useful tool, not just for field agriculture, but also for controlled environmental agriculture. Here we have the advantage of modulating the environment. By doing that, we can change different resource inputs to see the reaction of a plant phenotype within a particular greenhouse condition that could help to enhance the management practices. Now, as a CEA research lab, we focus on expedite biomass trait quantification using these digital tools. Also identifying more differences or earlier differences among treatments in a non-invasive way. However, for a production setup, we maybe are interested in a more general crop level growth monitoring and a yield prediction that could allow us to have a proper agronomic management based not just on environmental response, but also on the phenotypic response to that environment. Now, what are the possibilities with non-invasive imaging? We can describe dynamic plant responses by considering indirectly features from the plant morphology or from the plant physiology in reaction to different treatments. And additionally, we can add the analysis of multiple individuals as at an accelerated temporal resolution or at a high temporal resolution in combination with the environmental sensors that we can include into our calculations. Now, what do we do with all of that data collected? We are collecting data from images, we are collecting data from environmental sensors, but in order to streamline processes, what we want to do in research, try to make a substitution to the traditional destructive harvest we have, or if you want to improve your decision-making for your management practices within a greenhouse management, we need machine learning tools. Why machine learning? It's a pretty popular concept already, and we want to find patterns among the multidimensional or pluridimensional data that we have collected. Basically, even if you want to leverage your plant monitoring to improve that yield estimation, or even if you want to leverage the image processing to make it more efficient, to get results faster. Now, these are the three research hypotheses. I will not read all of them. I will just try to describe them pretty general. Chapter 1, we go into a tool to adapt segmentation into our variable image capture conditions within a greenhouse. On the second chapter, we go into estimating biomass, different biomass accumulation parameters. And then the third chapter, a little bit more applied, try to give some tools from a morphometric perspective and a spectral perspective to try to anticipate tipburn. Now, I will be quite general in the description of our materials and methods as we work with the same growing setup across the three experiments that I just described. Basically, this was the most adaptable setup. I mean, you can also consider it semi-commercial. So this was the most adaptable setup for us to consider our imaging strategy, which involved capturing multiple individuals within the same capture from a top view. We set the camera on top of all the trays that were distributed along a bench, and we captured an image for each individual tray in order to have a more high throughput analysis of our plants within a single image. Now, for experiment 1, we were not worried about the different treatments. We were more worried about producing different plant sizes to have a heterogeneous population of plants to describe better our model for biomass accumulation. On a second experiment, we added a calcium treatment because we wanted to figure out the right concentrations that we're going to be working with on the third experiment. Now, let's begin with the first chapter where we cover a reliable plant segmentation across the variable illumination conditions within a greenhouse. So Mark described it already. Basically, the segmentation purpose is to identify our object of interest within an image, in our case, our plants. So it's basically a classification task where we divide a pixel either as a plant pixel or as a background pixel. You can see there pretty clearly in the image. Now, this is what we want to achieve with the images we capture. However, we have our limitations in a semi-controlled condition where we're working now. Now, as imaging sensors, they also require good quality data as any other sensor, I mean. And basically, we require a good calibration of the sensor, but also, and more importantly, a correct processing of that information to gather that features per plant. So within semi-controlled conditions, there are specific factors we cannot control, like the sunlight hitting into our plants that produce different shades or brightness, or even algae growing on the background, could be either on the substrate or growing on your background, the background of your growing tray. That could affect the quality of the information you extract. You can clearly see here. And as I told you, one of the strategies is to set up the right processing pipeline, the right processing workflow. Because we don't only want to extract information from the pixels, but we want to give biological meaning to that information, right? So as you can see, improving that workflow, we can improve the quality of what we consider as a plant within an image. Now, threshold segmentation could be a simple. Segmentation could be as simple as a threshold selection of a particular color channel where you choose one single pixel intensity. It could be from the red, the green, or the blue color channel of an image to divide the pixels into what is an object and what is a background. We already went through that, but we are just considering a single criteria to decide what is a pixel or what is a background. Here, we want to introduce a model-based segmentation where we have different criteria. It could be different colors from the same image or even texture information coming from a color image that give us more criteria to improve the adaptability and with that, the processing automation of our image. Now, again, the objectives that would address the hypothesis I stated, we want to implement an automatic threshold. This is a little bit more advanced like the traditional threshold, and we want to compare that one as a baseline strategy to other two machine learning segmentation strategies. Mainly, we will evaluate that segmentation quality performance on unseen captures, information that the models haven't seen before, and with contrasting illumination conditions, but also with different plant sizes. So let's go ahead. I will quickly describe how the OTSU segmentation works. we basically started with our original RGB. We choose one color channel, could be from the RGB color space or any of the different color spaces that we know, to find the color channel that that shows the better contrast among background and a foreground or your object of interest. Then, as you have more contrast, it's easier to find that value of threshold. However, this Otsu segmentation automatically selects that threshold value without user intervention, where they can find the less within variation among a particular class and the highest variation among different classes. Then you can see you obtain your mask, but still we are struggling with the quality of that mask. Now, a supervised learning technique implies giving a model what you observe or what you are sure it's a plant object or a plant pixel. So let's remember that term, the pixel annotation. That's basically drawing what within your image is a plant. And that could be actually some handcrafted, it takes a lot of work doing that. So we constructed our models annotating each single images. We use in total 180 images. So we had enough information to track the models that we fit. And that's something I want to remember. The model's performance will depend on the reference quality. So we need to be really careful about how we annotate or label our images. Now, let me explain further this concept by explaining how each of the machine learning models work. So basically, to label our images, we use pre-trained models, segment anything model. It's a really popular modeling segmenting technique right now that works on pre-trained models. So we were sure that the information that we were providing as our ground truth information, as our observed values, have a good quality. Now, one of the problems with random forest is that we need to extract features. We need to extract that extra criteria that we need for the classification model. And something that's problematic with that is that those features are not always so adaptable, as we are changing constantly lighting conditions and so on. Now, after running the random forest models, I won't go so much into detail. We can discuss about that later. We obtain our mask based on the training images we provided at the beginning. So again, a key problem of the random forest is that feature extraction step, that it's really computer inefficient. Then we go for the unit segmentation. It's based on a convolutional neural network that it improves what the random forest wasn't doing. It is an automatically extraction, the most significant color or texture features that is extracted first in a general pattern and then in detail pixel by pixel to try to segment more efficiently what is a plant from what is not a plant. Now, how do we measure the quality among the different models? Well, we're working with classification models. Basically, we have 4 different performance metrics that, in short words, it's describing how closely the predicted segmentation matches the annotated mask that we discussed at the beginning. I just want you to remember what a false negative is. False negative is basically the pixel that it's considered to be a background, but it's actually a plant, and a false positive, the pixel that is considered to be a plant, but it's actually part of the background. So let's go ahead with the results. Now, the UNet segmentation was by far the best modeling strategy we could find for our conditions. As you can see here, this is the mask, what we were expecting from those predictions. Here we can compare the first approach, which is the threshold segmentation. As you can see, it takes a lot of the pixels of the background as planned, and it's not. Then the random forest segmentation really improves the quality of that segmentation by adding more color and texture information or further information from that image. But finally, the UNet segmentation that significantly improves the segmentation, as we can see based on this performance metric. But what we really wanted to do is actually adapt it to different illumination conditions and different plant sizes. You can see on top the original image. In the middle, you can see the reference or the annotated mask. And then in the bottom, you can see the results for each of the models. In this case, we're checking the Otsu segmentation. Based on the numbers and based on the image I'm showing, it didn't have a good performance. We go for the random forest segmentation. It significantly improves, there you can see in the bottom, the quality of the segmentation. However, the UNet segmentation is the one that gives us almost an identical mask, allowing us to keep the quality or of the canopy shape information that we will get in a future analytical step. Now, what we show here is that we kept the quality regardless of the condition. Basically, if we have high sunlight or we have dim conditions, we are sure that we will get the right segmentation quality to allow a better morphometric analysis of our shape features of the canopy. So basically, we are able to add that segmentation strategy across our growing setup, so we get good imaging quality results for further analysis. Now, conclusions, it's pretty clear. Unit strategy was the more efficient, not just for predicting pixels, but also for the training process. Then, as a semantic segmentation, that was an issue. A semantic segmentation just recognizes what is a plant or what is not. We still have problems with the overlap of plants. Mark already experienced that. We need to come with something else to try to avoid that overlap within plants, which could be maybe a strategy that works on instant segmentation, meaning that you recognize one single plant as an independent object, maybe in a later research. Okay, let's go to the second chapter. In our second chapter, we were worried now about estimating the biomass of lettuce plants. In this case, we want to integrate RGB and depth imaging. So here we wanted to obtain an accurate vertical height of the plant based on depth information. Then we wanted to generate an image processing workflow that would incorporate the information from the depth image and from the RGB image, so we can achieve a multi-plant feature extraction. Then we wanted to fit 3 different regression models using all the image-derived features that we did on the previous step to predict 3 different biomass accumulation parameters, mainly the fresh weight, leaf dry weight, and leaf area, what we are using in as a research CA research laboratory. Now, as I told you before, we were able to add into our image workflow the UNet segmentation to improve the quality of the information we are getting down the processing pipeline. So we are sure that we are getting, at least from the segmentation step, good quality definition of what is an object or what is a plant within our image. Now, how does the sensor that we use to get those vertical height information works? Well, it's a Kinect sensor, it's a depth sensor that besides capturing the RGB information, it adds an additional channel that describes the distance in millimeters for each pixel within the image. So it's the same resolution of image, but instead of having just 3 colors, it adds an additional dimension with the distance information. So that allows us, as you can see on the far right, to have a reference on how we can obtain the height for each single plant within our image dimensions. Now, what we did to train those models to predict the 3 biomass parameters more accurately? We extracted the geometrical features. If you don't know what a geometrical feature is, there it's a good description of what it is. It's basically the canopy shape description that it's showing us not just the area, but different geometrical parameters for that shape. Now, our aim was to predict 3 different response variables. So we needed to gather that ground truth, those observed values of the 3 predict biomass or the 3 responses. And we did that in 2 different harvest rounds as our observed information. So again, we're working with a supervised learning where we have labels, basically the observed values of our biomass information, and we have the predictors. The predictors is just the geometrical information and color information for each single canopy within our image. Basically, we have the correspondent label and the correspondent predictors for each single individual to train a model. Now we split our data, we split 70% of our data for training, and then we split 30% of our observations to test the performance of those predictions in unseen data. Now, the model comparison, we just, for the sake of comparison, we went ahead and worked with a simple linear regression model at the beginning by choosing the best correlated parameter that we thought might be the area. in pixels from all the geometric information we extracted. In a lasso regression, it's still working, it is still a linear regression approach, but it's regularized, meaning that we are adding a penalty that rejects useless coefficients from all the variables that we use. Finally, we chose a random forest regression approach to account for those nonlinear relationships that we could find among all the 12 parameters that we used. And we compare all the three models on how they perform in each harvest round. So basically, one model per harvest round, and for each of the responses we tried to predict. Now for the results, you can see here how accurate were those height measurements of our plants. Basically, the best reference that we could find was the maximum height of the pixels around the plant that allow us to get values of plant height compared with the observed values with the ruler with a 1.1 centimeters of difference. That is almost, it's an acceptable error, a low error rate. Now, what's interesting is that in our second experiment where I told you that we tried calcium concentrations, obviously the treatment with the lowest calcium concentration show a tipburn phenotype, where we had a reduced ground and it was pretty visible with the height of the plant. And our sensor was able to follow that pattern of the ground of the plants that were under calcium stress, under calcium deficiency. There on the left, you can see the box plots for the actual height or the observed height value. Then on the right, you can see the image derived height values that actually follows a similar pattern. Now, we analyzed the correlations among responses and among features to set the variable that better correlates with the leaf fresh weight and all of our response variables. And we also evaluate the correlations among the predictors we used. And we found a warning here, which is the multicollinearity, which actually makes sense as we're extracting variables from the same shape, from the same image canopy. We were expecting to find some correlations between those features, those geometrical features. However, for the leaf fresh weight, if we compare among models based on three different performance metrics, the R-squared, how much of that variability in the response was explained, then the root mean squared error, meaning what was the rate of error given by that model, and a percentage description of that error, we can see that the lasso outperformed the other two models, not just for plants on the beginning of the first harvest round, but also for plants of 19 to 28 days old for a second harvest round. Which that one was the challenge as the plants, the older they are, they start to get the influence from the different treatments we apply. So it's more challenging, not just from the treatments, but from the environment. So it's way more challenging to predict that growth. However, the lasso was the model that was able to predict or explain that variability in leaf fresh weight with a 70%. And then the error in grams was of 13 grams. If we consider that the medium size at that stage was 113 grams, that represents an 8% error. We did the same with the leaf dry weight. Again, the lasso regression still outperforms the other two, even the more complex strategy. And then for leaf area, some of my colleagues will be happy to see that leaf area can be actually predicted by imaging techniques. But here is where the model performed again, lasso model performed the best with the better predictions with error rates as low as 8%. And what's interesting about lasso is even though it considered all the features together at the same time, it has a penalty added, and it rejects the coefficients that do not contribute to the explanation of that variability, just leaving us with the coefficients that really contribute to the model predictability. So then the conclusions for this one, we have a depth sensing that provided accurate height measurements that yield a better plant structure. So we went from a 2D plant structure to a more volumetric, more real plant structure description. Then the predictive models improved the biomass estimation using both color and morphometric description. Then we need to keep in mind that not always the most complex is the best, especially now that we use one single sensor to analyze the whole information. So basically one lasso regression that is still based on linear regression outperformed more complicated models like the random forest for all biomass parameters considered. Now, the same model, the lasso regression, tell us something about the importance of consciously selecting what are the most, or what are the features to be selected before feeding a model. Now, the final experiment that we had an intention to be like a more applicable experiment using the morphometric description that we could gather from our image processing pipeline. And we wanted to provide some tools from a morphometric analysis and from a multispectral analysis to try to monitor tipburn development. Now, the tipburn is not just, as you might consider, lack of availability of calcium within the nutrient solution, but it's more an interaction with the environment, an environment that drives an accelerated growth rate, limits the mass flow of calcium to the young tissues of the plant, and that's why it shows the particular pattern of necrosis and reduce a rate specifically in the center of the canopy, right? So there are some solutions right now, environmental modulation, again, adding airflow or considering a clear cultivar selection that sometimes it doesn't correspond to the description of the manufacturer. It says tipburn tolerant, but it's not so tolerant if you are using LED lights on top. Now, there's a potential of applying products within your nutrient solution that could help you avoid this problem within plants. Exogenous melatonin is one of the biostimulants or that we tried to add into our system to avoid the development of that tipburn. And why we thought that this might work? Well, it has been shown in different reviews that the physiological mechanisms related melatonin acts in different physiological mechanisms related to stress tolerance. As you can see here, it's a broad spectrum antioxidant, and it also helps with the gas exchange, improving gas exchange parameters within the plant. So we thought that melatonin could be effective to tipburn as the reactive oxygen accumulation and tipburn development are quite connected. Basically, there was a study that linked the oxidative stress and the tipburn development by showing how in different cultivars or cultivars with higher antioxidant activity were able to mitigate the ROS or reactive oxygen species damage. And hence they show less tipburn susceptibility. Now, we were saying that melatonin is also helping with the gas exchange parameters. Basically, if we improve the transpiration or the mass flow of that particular plant with the stress, we fight against that low evapotranspiration environment that is restricting the mass flow, and it's provoking that limit calcium mobility. So besides that, we wanted to describe the tipburn development through a broader spectral information. So for now, we work with green, red, and blue color channels, but we wanted to go broader into broader regions of the spectrum to try to anticipate specific physiological changes. So the reflection of certain colors are influenced by the physiological processes that are not visible in the spectrum of light. So we need to differentiate between multispectral and hyperspectral. Basically, in our setup, we will consider just multispectral analysis where we have a several description of pre-selected bands instead of a continuum of the spectrum, limited bands, in fact. So for this, we set the following objectives. We wanted to evaluate the potential of using morphometric description and also a spectral description to anticipate leaf tipburn in lettuce. Then we wanted to assess the effect of combining image and environmental sensor features to improve estimation models performance. So with this, we want to complement the morphological features that we already extracted on the previous experiment with more physiological description of the plant given by the reflectance that give us more criteria actually to anticipate a specific physiological response. So that would be the recipe for an early stress detection for the tipburn. Now, would it be effective? Well, In our experimental setup, we had three different treatment factors where we had different light levels, different melatonin concentrations, and different cultivars within the same growing setup. Now, as you can see, a few of the traits were growing on top of the supplemental light that, of course, induced the tipburn development, and some were growing just under sunlight within our greenhouse. Now, we were having problems with the overlap again. Mainly when the plants were touching each other, we had problems selecting individual plants. The same happened even though we harvested plants to create more plant space among individuals, but the same problem still happened. We were unable to identify individual plants. So we came with another, even though we proved a quite effective strategy for segmentation. Now, instance or identifying individual objects within the plant, within the growing setup might be the right solution, where regardless of the intimate touch of two plants, you are able to separate two individuals. Now, I won't go further in this, but that allows us to get localized information for each single plant. Now, another thing that we were struggling with in the previous experiment was multicollinearity. That basically we're extracting really similar information from the color images or the shade properties. However, we can enrich that information, that geometrical information, by combining features. Basically, features coming from the light reception, from a light sensor, from a quantum sensor, maybe with the area that the plant actually, Mark van Giersel was working really broadly with the concept of incident light per plant. And we wanted to combine all the area measurements that we captured per canopy with the readings that we have in each independent tray. So each tray within our setup had one single quantum sensor that gives us information of the cumulative DLI that the plant had until a specific point in time, giving us an incident light per plant. Obviously, we're assuming that the distribution of those LED lights is homogeneous across the whole LED, but it's still a localized measurement of the light across the tray. So let me talk a little bit about the Rain multispectral camera system that we use for this setup. This is a lighting company that developed this multispectral setup that uses a monochromatic camera that actually use an active light source illumination, LED lights that are turning on one at a time to capture the information or the spectral information coming from 9 different mean peak wavelengths or bands. So with this, we're able to construct not just a 3-dimensional color information, RGB, but we can add image cube. What I mean by image cube, basically the spatial information besides the spectral wavelength information that complements our analysis. Now, using that information, that reflectance information, we can have 2 approaches. Basically, capture the reflectance coming from each specific band. It would be the mean reflectance for each single band, or we have the options of using vegetation indexes values. So this is maybe a safer strategy as we calculate these based on 2 or more bands. One of those bands is the measuring the measuring band where we want to grasp information from, and the other one is just a reference band that actually accounts for the effects of the non-related reflectance of a particular band. Now, here I'm using the example of the photochemical reflectance index, an index that is really correlated with the Santofield cycle that gives us information basically of the light use efficiency that a plant could have. Now, let's go faster through the results. Let me disappoint you a little bit. The melatonin didn't have any effect on plant growth. That was our first analysis. If we could see any effect maybe on the plant growth, as all papers have shown, maybe there's something else going on the plant. We didn't see anything, even though we capture information across time for every single day of the crop cycle. There was not a particular growing pattern under different melatonin concentrations. So I would suggest here trying to extend that dose response curve as we just tried 3 different concentrations of melatonin. We were not sure really if this work under those concentrations, even though we tried higher concentrations that were detrimental for the plant. Now, I want to highlight this slide, considering that for the nutrient concentration in the table in the bottom, for all 6 trays, we have the same calcium concentration for the 6 of them. However, on top, we can see the DLI, the summary of the DLI for all the growing trays. And as you can see in red, the plants, the trays that grow under supplemental light had a considerably significant DLI from the plants that didn't grow with a supplemental light on top, and only the trays with the higher DLI got affected by a tipburn development. So basically, the tipburn was not related to the calcium concentration, but instead to an environmental response. Now, let me disappoint you one more time. Melatonin didn't have any effect on the tipburn development. We didn't have any clear pattern, even though it seemed that the 5 micromolar melatonin had some effect on reducing the severity, but it's not clear, it's not significant. I'm showing here the coefficient, the confidence of interval at 95% for all the graphs that I'll be showing. However, the morphometric changes across time were giving us important information about those changes in light. Here we are not considering anymore the different melatonin concentrations, but rather we are considering the other 2 growing factors, cultivar and supplemental light. And as we can see here, 2 of the geometrical descriptions that we use for the plants show a clear difference pattern of plants growing without light and for plants growing with supplemental light. There you can see the compactness and the extent within the dash square. Compactness is actually just a proxy for compactness from an image analysis perspective. Then extent give us also an idea of how much the canopy expanded under certain conditions. Now, if you consider the graph under the supplemental light, well, the compactness of both plants change, but specifically the plant that was more susceptible, this is cultivar dragoon, was even more compact than the less susceptible chicarita. So we can see that besides plant area, there are morphometric descriptions that can correlate with that response of the plant morphology to the light conditions. Now, I want to go again to the models that we fit on the second experiment, but I would like to add the significance of the incident light parameter that we use to calculate, that we use area and the quantum sensory readings to set these new parameters. So incident light, it doesn't improve so much the correlation between leaf fresh weight and incident light or leaf dry weight and incident light. However, if we use a multiple linear regression that includes incident light and another of the geometrical features, we can see how it outperforms a multiple linear regression that just uses area. What I'm trying to convey with this is that actually just incident light gives a broader or more information, helps us explain more information of the variability of in this case, leaf fresh weight by adding that sensor information within the image derived frame. Now, let me go, I'm almost out of time, but let me go pretty fast with how we perform that multispectral analysis. We basically capture images all across the growing cycle. However, as we wanted to anticipate tipburn that happened around 12 days after transfer, we wanted to consider just the first 10 days images, basically to see the effect on the reflectance of the plant. You can see here the pipeline that we follow, it's similar. We want to isolate the plants first. We want to delimit the area. In this case, we want to delimit the area. We are not considering the whole tray as we were using as an active illumination source, the LED lights coming from the same sensor that didn't have such a good distribution across our the whole extension of our tray. So with the imaging, we limited that analysis to just 8 plants per tray. We had 4 per each cultivar, but at least we make sure that all those plants had an even LED light distribution, which actually the images were captured at night where none influence from other light sources was evident. So the only reflection that the sensor was measuring was the LED being reflected from the plant canopy. Now, the first analysis was the mean reflectance per band. Here you can see from left to right the whole spectrum that we considered, going from the visible spectrum, where we actually saw quite a significant difference among Chikorita and Dragoon. Well, actually, those differences were expected. You could see that one cultivar had a darker green color than the other. And however, we could also attribute this to a more efficient blue and red light absorption, as they didn't, especially for the plants that were growing under supplemental light, as they were not reflecting, or the plants that were not growing under supplemental light were not reflecting were reflecting more green, blue, and red spectrum, meaning that they are not absorbing as efficient as the plant with supplemental light on top, maybe because plants were able to develop more chlorophyll with the supplemental light. Now, there were no significant differences among the mean reflectance per band for the red edge or near infrared part of the spectrum. However, it's really interesting to analyze from a spectral vegetation indices perspective by cultivar. Again, we could attribute this to the particularities of each cultivar, where actually these indexes, the three of them, are related to how the greenness of the plant or how much chlorophyll could have been accumulated by those plants. Of course, these are not used independently. They are usually added into models to try to predict that chlorophyll correlation, but that's what it's usually more correlated with. And in fact, in orange, you can see the Dragoon cultivar that had a darker green, and that's why it has a higher index values for these specific indexes we consider. Now, it's really interesting also to see what happened with the photochemical reflectance index for the plants growing under different supplemental light conditions, with or without it. Basically, in the photochemical reflectance index, we can see that the plants growing with supplemental light on top had a higher value, in that they are having a better light use efficiency, as we are providing more light to them, even though it's not a limiting quantity of light that we are providing. It seems that they are making a better, a more efficient use of light under supplemental light. Now, the chlorophyll index is again related to the chlorophyll prediction of chlorophyll content in plants, and maybe the amount of light that the plants receive could have impacted the chlorophyll content, and thus give a higher value for the plants that grew under supplemental light conditions. So to conclude, adding a broader morphometric description helps characterize the morphological changes to light that could explain why tipburn or a specific cultivar is more susceptible to tipburn. Now, if associating localized environmental information with sensors, environmental sensors, with remote sense plant description, basically what we got from the images, allow us to create features or traits that improve the predictability of growing models. Then to actually predict or earlier predict tipburn, we require multivariate data analysis or another machine learning tools that could let us associate morphometric information and indices values and mean reflectance as a better tool and a better association of those indirect measurements. So without further ado, I just want to acknowledge all my lab members, the Thesis lab members, the Katrina Research Institute, the people who help us with a few of the tasks of the image analysis, all my committee members and collaborators, and a special thanks to Dr. Alexander Kushera from the RAIN team, who gave us the backup with the multispectral information. Read the reference, and thanks to my amazing team. Any questions from Zoom, maybe?
Could you speculate as to what concentration of melatonin might be effective. I know you tested a high concentration and then your concentrations that you presented were too low maybe to have any effect, but where do you think the middle ground might be?
I would say, so we tested before in a preliminary experiment concentrations between 60 and 600 micromolar. Here we went from 5 to 10, so we were limited in extension. However, with the 60 and 600, we saw a considerable detrimental effect
At 60?
at 60 and still at 60, and at 600 was even worse, so
Yeah.
basically.
Yeah, so somewhere
I would
between.
say we are close.
Cool.
It could be under those 50 micromolar. So there's an interesting meta-analysis that I cited here that shows where that concentration is more effective, but again, depends on the cultivar or the species you are considering for your analysis, and more significantly, the application method. There's a lot of research to be done about that, but maybe try to localize there's not so much research done in lettuce specifically for this type of stress, but probably trying to look for the right concentration might be looking for a more pronounced stress condition. Go ahead.
So Jonathan, looking in perspective, I don't know what we were thinking, because people use melatonin as a sleeping aid. Were we thinking that plants were going to be more active? Taking calcium, so I think we should have revisited, that a little bit.
But actually makes sense with the reactive, the antioxidant capacity that it has shown in different studies, and also improving the gas exchange. But it has been significant in type of studies. It's a good option, Go ahead, Chris.
When you applied the melatonin, I can't remember, was it just at the beginning of the experiment?
Yeah, actually,
Single dose and then.
I didn't explain that in fact. And what I did, I added powdered melatonin within my nutrient solution in each single tray at the different concentrations. I will dissolve the melatonin in a separate volume of water, and then I will dilute it within my tray according to the concentration I wanted to add it to each tray.
Do you have any thoughts on like the integrity of the melatonin? Because it seems like at the end of the trial, when the plants are really the most susceptible to getting tipburn,
Yeah, actually,
it's still like in an adequate concentration.
Actually, there are many studies that prove that they test with implant melatonin concentration after application. That actually could be a good method, but even we can, I don't know if we could test it also within the nutrient concentration, but yeah, maybe in tissue would be our direct, could be helpful to explain if it is actually absorbed by it. Go ahead.
Yes, back to imaging. So when I started to be intimate, you had a really good model. So is it working throughout the whole growth cycle, or is it there at a time where it's like, I don't know, this still looks very similar?
No, actually, luckily it worked through the whole crop cycle. The challenge is that as I was working with 3 different harvesting rounds, I was always giving a little bit more space, even though they were touching each other, but they were not like so cramped together that, but it would be interesting to try in like a cramped space.
Can help you.
Yeah. Any other question maybe for Sam?
All right. Thank you very much, Jonathan. Thank